Let's begin with the controls used to license each clinical trial for every routine injected childhood vaccine. Again, controls are critical because if you don't have a baseline of safety in your clinical trial, your comparison may not be appropriate when it comes to safety. It might be for efficacy, but not for safety. And so for the two current standalone hepatitis B vaccines, Engerix B and Recombavax HB, what you'll see is that they were licensed for children based on an uncontrolled trial. So there was no control group in the clinical trial relied upon to license those for children. What they were comparing it against is unknown. For DTaP, there are two licensed standalone vaccines, Infanrix and Dapticel, both licensed based on trials where DTP was used as a control, but DTP was never licensed based on a placebo-controlled trial. The three Hib vaccines you can see on this chart again when you go down the chain, ACTHIB, licensed against the Hep B vaccine, which itself was not licensed based on a placebo-controlled trial. Same thing for Hibarex, ACTHIB, then Hep B, then no control, and PEDVAX-HIB, which was licensed against where the control was a lyophilized PEDVAX-HIB, but the control group in the trial relied upon for that vaccine was an injection that included lactose, aluminum adjuvant, and thimerosal all in the vial. Again, not a placebo, not inert. And then the two PCV vaccines, you can see the chain as well. They were both licensed based on trials where Prevnar 13 was used as a control. Prevnar 13 was licensed based on a trial which Prevnar 7 was used as a control. And Prevnar 7 was licensed based on a trial in which another investigational vaccine was used as a control. Prevnar 7 being the first licensed PCV vaccine, there was certainly no excuse to not use an inert control for that trial. IPOL, which was licensed based on a clinical trial with 3 days of safety review and no control group. It was licensed in 1990. This is not the same product that preceded it from 1955. The Salk vaccine, which was removed from the market in the 1960s. This one was made with Vero cells. It had 2, 4, or 8 times the amount of antigen, depending on whether you're talking type 1, 2, or 3. Had a different formulation, different manufacturing process, completely different product. At least OPV could have been used as a control, presumably, but that had no control. And then there's various influenza vaccines. I will not go through those because there are many of them, but they will be detailed in the memorandum that I'm submitting that will accompany this, that will include the citations for everything on some of these slides. Most of the citations are at the bottom, but for some of them, they were too voluminous. They needed to be put in a separate memorandum. And then on this slide, we're going to go through all of the vaccines given 7 months and recommended for routine injection 7 months and older. Again, these are all the routine injected standalone vaccines for 7 months and older on the CDC schedule currently. And as we go down the list, starting at the top, we've got MMR2, which was licensed in 1978 in a clinical trial that had less than 1,000 children, reviewed safety for 42 days. It was only 1 dose. And there was no control group. They could have presumably used the MMR1 was licensed in 1971, but they did not. They could have maybe used standalones. They did not. There was no control group. And it's a very different product, and it very much needed its own. It should have been done if they thought that MMR1 had a proper safety baseline. Unlike the prior versions, for example, each vial of MMR2 contains, because the rubella component was now was grown on the cultured cell line of aborted fetus, includes billions of pieces of DNA from that cultured cell line, human DNA in each vial, which was not true of the prior vaccine. There are other major differences between the MMR2. It should have been properly trialed. There was no control in that trial, and it was underpowered anyway, and reviewed safe 42 days. And those trial reports I think are fascinating. Folks should look at them. About 1/3 of the kids had gastrointestinal issues. 1/3 of the children had respiratory issues. And there were other complications, but since there was no baseline of safety to compare it against, unclear how the FDA determined it was safe. Then Priorix, which is more recently licensed MMR vaccine, you then used MMR2 in its as the control. And actually, when you look at that clinical trial, and you look at the amount of adverse events, serious adverse events, as well as new onset medical conditions, in the 6 months that they finally did review safety in that trial, they were similar, but they were, I would argue, concerning. But because they were similar and MMR2 was presumed to be safe, it was not viewed as a concern. That brings us to Verivax, the very first and ever chickenpox vaccine licensed in the United States. No reason to not use an inert control group, but instead there were only a few hundred children who did not get an inert injection. They got an injection of 45 milligrams of neomycin, which is an antibiotic, which is per milliliter, which is obviously not inert. Topically, it can cause complications. Then you have two hepatitis A vaccine, Havrix and Vacta. They were both licensed right around the same time. No existing hepatitis A vaccine. No reason to not use an appropriate inert control to license those. But yet Havrix, for reasons that I cannot explain, had Engerix B as the only control that can be identified in one of its small trials. And Vacta was licensed based on a control group that received an injection of AAHS. That is Merck's proprietary aluminum adjuvant that scientists still are unable, independent scientists cannot get hold of to actually test, and thimerosal in the vial of each control. Why you would inject anybody with two substances that have no known therapeutic value when injected on their own is, I don't understand, and I don't even know how it's ethical, but yet that is what the control group received, again, not providing a proper baseline for safety. Then we have the two Tdap vaccines that were licensed in the U.S., Boostrix and Adacel. They used Decavax as a control. Decavac is only licensed for adults. Decavac was licensed in a trial with no control group. And then we have Gardasil 9, which was licensed in a control, based on a clinical trial in which Gardasil 4 was used as the control for almost everybody, except for about 306 subjects who got did get a placebo, finally, an actual placebo, so you can use one, but only after they got 3 doses of Gardasil 4. And Gardasil 4 was licensed based on a trial in which almost everybody in the control arm was injected with AHS, the aluminum adjuvant, as well as the carrier solution. Again, not sure how that's ethical, but that's a separate issue. And then a few hundred got just an injection of the carrier solution, which is labeled as a saline placebo in the package insert, interestingly, but in the underlying documents is clearly not. It included everything in the vial except for the antigens and the adjuvant, including the yeast protein and so forth. And finally, and I hope I'm not putting anybody to sleep, brings us to the meningococcal ACWY, and there's currently 2 on the schedule, Menveo and Menquadfi, and when you go down the chain of those, Menveo was licensed based on a trial in which Menoctra was the control, Menoctra of Menomune was licensed without a control group. Same for the other one. Interestingly, today, if you pull up the package insert, and as required by federal regulations, Section 6.1 is supposed to reflect the clinical trial relied upon vis-a-vis safety for that product. And interestingly, the Menomune and the Menoctra package insert, those are the 2 oldest ones, both have the same clinical trial listed in Section 6.1. The clinical trial relied upon to license Menoctra, in which Menomune was the control. So apparently Menomune, Section 6.1 refers to a trial in which it was a control in its own package insert. Obviously, it didn't have that initially because that trial didn't exist. Okay, that takes us through every single standalone routine injected childhood vaccine on the schedule. And as you can see, the concern is that not one of them was licensed based on a placebo-controlled clinical trial, nor was any vaccine used as a control to license any of those vaccines, licensed based on a placebo-controlled trial, nowhere down the chain. I know there's a lot of folks in the press who are here, and they might go ask what they would call experts about this question. What I would suggest to them very strongly instead of asking people to look at the primary FDA sources yourself. If you want to know what was relied upon to license a vaccine by the FDA, ask or look at the FDA documentation. And the memo that I'm going to be, I've done this many times already, it's all available. It's on the FDA website, actually. So you could just go to the FDA website, or you could look at the supplement filing that I will be providing. And I suggest you look at that, in which I will detail and list every single citation, which already might be on this AC website or it's in the works. Okay. And we shouldn't even have to do this because the Secretary of HHS has made this point clear many times, and it is in fact a fact. And it's not just it really is an important point. It's a critical point because without a proper baseline of safety, you're laying assumption upon assumption that the product is safe as you see serious adverse event rates in those trials, because you're just looking to see is as safe as the existing product. And we'll look at one or two examples